All right, in our continuing saga for the search for DNA, we still need one more experiment to really convince scientists. Here's an example of sometimes we can be pretty stubborn and we have an idea and we want to stick with it. And the idea is that, we really did not necessarily believe that DNA was genetic material. Now, people were getting pretty convinced. Avery's experiment was pretty convincing, but I think people were still wanting to know for certain. And so, notice the year 1952. We're still kind of taking a long time, and part of this is the technology. So, a little bit of background here. What we're going to use is a virus, but this is a virus that infects bacteria, and so they're called bacteriophages. And up here on the picture, you can see what it looks like. Here we have bacteria that is coated with bacteriophage viruses. Now, so these guys are harmless to us, but they do have a pretty wild appearance. They look like little spaceships or hypodermic needles. And now we know that what they do is they have DNA inside a protein coat and they inject DNA in. But remember, they did not know this in the 50s. They knew that they infected bacteria, they had isolated them, but they weren't sure how they did it. So remember our question, is DNA the genetic material or is protein the genetic material? And so, if you wanted to know which one changed cells, you might use a molecule that is made only of DNA and proteins. And that's what they did. And so along comes Alfred Hershey and Martha Chase. Hey, a woman, finally. So Alfred Hershey and Martha Chase. And what they're going to do is use a bacteriophage, which is going to be made of a protein coat. Now remember, proteins have sulfur, but they don't have any phosphorus. And then the DNA would be inside. The DNA has phosphorus, but no sulfur. And so what they came up with was a pretty clever way to use that to our advantage. And so you can imagine the idea being is if you were trying to figure out which one infects cells and causes genetic damage, what if we could label them somehow and follow where they go? And so what they did is they actually made two different batches of bacteriophages. And they used isotopes that were radioactive. And so these isotopes do exist. One of them is a radioactive isotope of sulfur called sulfur 35. And then another one is a radioactive isotope of phosphorus, phosphorus 32. And so what they did is they took batches of virus and bacteria. Viruses, you need to have bacteria to grow them. And so you grow viruses in a medium that has radioactive sulfur. It would be like if I fed you glow in the dark chocolate chip cookies and you ate them and suddenly you glowed in the dark. And that's the idea. You're going to incorporate the chocolate chip cookies and suddenly that glow in the dark is in you. And so that's what they did. They made viruses. They made two batches. They made one batch that had radioactive sulfur. And remember, that means their proteins are going to be radioactive. So along comes a radioactive bacteriophage, and I colored it pink. Yay. Okay. And so this is now a radioactive protein coat. The DNA, notice I left black. It is not radioactive. But the second batch, they grew in radioactive phosphorus. And so in that regard, only the DNA is going to be radioactive and the protein is going to be black. It's not radioactive. And so now you've got two batches and you've got a prime example of an experiment where we're now going to track what happens. We're going to follow the radioactivity. And it's pretty darn clever. And this really was the experiment that kind of settled the question. Now, for all of the elaborateness of growing the bacteria with radioactive isotopes, I always find it a little amusing that in the next picture I show a good old kitchen blender. Because it's certainly not the most impressive of lab equipment, but that's what they did. So we've got ourselves the two batches, and what we're going to do is let these radioactive bacteria infect. So that's the first step. We're going to let them infect the bacteria. And remember, whatever is being injected into the bacteria is the genetic material. And we're trying to find out what gets injected. And so we're following the radioactivity. Now, in the first example, the protein is radioactive, remember. Now, if it's the protein that's if it's the protein that's being injected into the bacteria, we would expect that the radioactivity would be inside the bacteria. Uh, and so what they did is they took all the cells and they put them in the blender and they just pulsed it very quickly. And what that does is it shakes off all of the virus particles. I know, kind of weird, but again, here we have our humble blender. And so we're going to shake off our viruses. And whatever is inside the bacteria, remember, is what gets injected and what is infecting the bacteria. Well, then they centrifuge, and remember, a centrifuge is the machine that spins around and it pulls the heavier materials down. So the heavier cells are going to be at the bottom, and the liquid is going to be at the top. Now, remember, the bacteria cells are going to have the injected material. Now, we know it's DNA because we know that DNA is what is the genetic material. But remember, they didn't know that at the time. So they're trying to figure out, would the protein be in the cell or would the protein be in the liquid? That's kind of their question. So they took the mixture and they centrifuged it. And what they found is that the liquid was radioactive and the cells were not. And so what they realized is that means the radioactive protein is not in the cells. And so their conclusion is that the protein is not going into the cells. The second batch, they made the DNA radioactive. And so they repeated it. They let the radioactive viruses infect the bacteria cells. They then put the whole thing in a blender, just shaking them off. And you got to keep in mind that the protein coat is not radioactive. It's the DNA. And so notice the radioactive DNA went into the cells. So this time, when they centrifuged it, the radioactivity was down at the bottom of the test tube, and the liquid is not radioactive. And so what they realize is the radioactive DNA is at the bottom of the test tube, and the radioactive DNA is at the bottom of the test tube, and therefore the conclusion is the DNA is going into the cells. Sorry, let me change that real quickly. The DNA is going into the cells. That's what happens when you copy and paste. So let me make sure that that is accurate there in my video. So the DNA is going into the cells, and that was kind of the conclusive proof that the DNA is the genetic material. That's what finally kind of convinced everybody. So there we have Hershey and Chase together taken outside their research lab. And that is, again, the last kind of proof that DNA is the genetic material. Now the question becomes, how does DNA work and what is its structure? And for that, we're going to watch another video that kind of shows that history.